and welcome to episode 24 of the Breaking Bio podcast. My name is Stephen Hamblin, postdoc at the University of New South Wales in Sydney, Australia. Today is Thursday, March 28th, and even though we're completely off the rails, we're struggling forward with the show here. So we're going to start with the usual menagerie of introductions.
Hi, I'm Rafael Maya, Ph.D. student at the University of Akron, Ohio.
Hi, I'm Morgan Jackson. I'm a PhD student at the University of Guelph in Ontario, Canada.
Hi, my name is Heidi Smith, and I'm a postdoc at the University of Texas at Austin. And we are joined today with the newly minted Ph.D.
Hi, I'm Anthony Salvagno. I'm newly minted Ph.D. at the University of New Mexico.
Yeah, so you had your PhD defense this morning, right?
Yeah.
And you passed.
I did pass. So people watch the Hangout.
Yeah, no. We typically have like one viewer, but that's all right. I was waiting for a chance to use those Google effects for some useful reason. So how was your defense?
It went really well. I presented for 50 minutes, and then some people in the audience asked me questions. One of my friends actually asked me a question on like some photosynthetic pathway that I have no idea about, because I'm not a biologist and I'm not a plant scientist, even though I did present a little bit about plants.
Because some of it's like biology and physics, and yeah, this girl asked a question about C3 versus C4 plants.
And I just stood there and I was like, I have no idea what you just asked me.
So, well, why don't you tell us a little bit about your research, just so for the people at home and to bring us up to speed.
Yeah. So I presented my dissertation today about the effects of heavy water on living organisms. And in case you don't know, heavy water is made of the heavy isotope of hydrogen, which is called deuterium, and it's D2O instead of H2O. And so it has a 10% mass increase over normal water. And there's a lot of interesting effects because of it. Basically, if you have high concentrations of heavy water, living organisms will react slower. They'll grow slower. They'll be toxic to most higher organisms like plants, animals. But there's also some really interesting effects at low levels of deuterium around between natural concentration amounts, which is 0.01% and 1% D2O. And so that's some of the stuff that I presented about today. Basically, you can have increased growth in higher concentrations of D2O. Sorry, in low concentrations, but higher than natural concentrations.
Well, I can see, I'm looking at some of your defense outline from your open notebook, which we'll get to in a bit. But I see you're talking about Arabidopsis and E. coli and yeast.
Yeah, I think it's the most organisms I've seen span in someone's defense talk. Because
So
all the way from E. coli to, I guess the tobacco plant is the biggest one, right?
Yeah.
The most higher up on the evolutionary tree.
I didn't report on any of the stuff that I did on myself.
Yeah, you're going to save that for your second career in doping athletes with deuterium.
Yeah.
So, what kind of size of effects are we looking at here? Like, are we talking monster Arabidopsis or?
No. So the most in depth that we did was we actually studied like the growth rate of roots for tobacco seeds, and we found that it was like on the order of, I actually don't even remember the number. I'd have to look at my open notebook, but which is now more accessible to you than it is me for some reason. If I recall, it was like it grew like 8 millimeters in 10 days in 1% D2O, and it grew like 5 millimeters in the same time span in normal water. So roughly like 3 millimeter difference, which is pretty dramatic. And so like if that extrapolates over the growth of the plant cycle, the plant would probably be bigger, where it would have at least achieved its lifespan in a shorter amount of time.
I thought the more like striking, because I thought initially you'd see more striking things from plants, but it was actually more amazing to look at E. coli. Because just cell growth, like the cell morphology changed so much.
How so?
In E. coli, the cells, we didn't actually quantify this, but the cells looked like they were probably about double the size in D2O after they were adapted to D2O than they were in normal water. And by D2O, I mean we did like experiments with pure heavy water, which is normally toxic to higher organisms, but to E. coli it was actually not all that toxic.
And they grew more then? just in pure heavy water?
They wouldn't grow more. They grew about as good as they would grow in normal water. But after they became adapted to it, they could grow equally well. And then if I put E. coli from the adapted E. coli from D2O into normal water, they would like grow like gangbusters.
Oh, you should be talking to Richard Lenski, it seems.
Yeah, it was almost like if you look at the results part, when you looked at like E. coli, yeast, or whatever, like D2O versus H2O in different concentrations, it was like each one of those to study the effects could be someone else's PhD project.
Yeah, I kind of just did like basic research in a whole bunch of different fields with the expectation that like experts in each field could pick up on the research and go on from there.
Fair enough. What led you to this? I mean, was it just for the sheer unadulterated science of it, or is there something specific you want to get out of that application of D2O?
So originally it was kind of two things. One, it was like, this is a super fun project that I could do. I like to do like lots of different things in the lab, so this was like one way that I could be really diverse with my skill set. But then also we had this grant to study solvent effects on kinesin, which is a molecular motor. And so the solvent that we were playing with was water. And so one of the simplest things we could do is just change H2O to D2O. So a big portion of the lab was funded to do that kind of research. But we came across like the history of the experiments. And it was basically like Gilbert Lewis, who first purified heavy water in 1933, did some basic experiments where he put tobacco seeds in heavy water and found that it was toxic to higher organisms. And then a whole bunch of other scientists came along and did kind of the same research. But the initial question was, is heavy water, this was proposed by Gilbert Lewis, is heavy water merely tolerated by living organisms, or is there some evolutionary use for it? And so no one had ever answered that question when it was first asked back in 1933. And so it seems like there was this huge gap in research where everyone just went off and did this one thing, but nobody had ever answered this really fundamental question. So it was like, I get to have a lot of fun in the lab, do some really basic but fundamental research, and then also get to learn a lot about the history of these projects.
Nice.
So maybe you said this and I didn't pay attention, but so do you find like this heavy water naturally, like in small concentrations, like everywhere, or only in restricted places?
No, so like there's a term called standard mean ocean water, and it just has like, if you go to the Wikipedia article on it, there's just like a breakdown of all the isotope ratios of different atoms in water. And so the ratio for deuterium to hydrogen is 1 in 6,420, 6,420 atoms. So one of them is deuterium. And I think that's pretty standard across the planet, but different areas may have different isotopic ratios. And so one of the things that I presented on today was that we actually came up with like a relatively cheap way to characterize the local isotope ratio. Basically, like if I gave you a milliliter of deuterium depleted water, which has no water in it, and I gave you, no deuterium in it, and I gave you a milliliter of water that was spiked with heavy hydrogen and heavy oxygen, both of them, like through atmospheric contact, will start to regress back towards natural water concentrations. And so we can track the regression and pinpoint what the local atmospheric isotope ratios are.
It's just that Heidi mentioned something about it being really expensive. So, like, do you synthesize it, or do you have to extract it from unusual places?
I have to buy it commercially. I have no idea what the equipment cost is, but I know they use like super huge equipment to purify it, because it's mostly used in like nuclear reactors as a neutron moderator.
No.
Oh, neutron moderators are so pricey, you know?
Oh, the neutron moderator.
How much is the heavy water? Like, what are we talking?
So for 100 grams of heavy water, it cost me $80, and that's at the university discount. And interestingly enough, deuterium depleted water, which is literally the exact opposite of heavy water, is $120. And I think that's a huge ripoff, because I'm pretty sure they both come out of the same machine. It's like you have all this extra water left over that's not D2O. Why are you charging me more for the pure water?
And it's like diamonds. They keep it all in a safe somewhere, and they're like, oh, yeah, this shit's abundant. Drive out the prices, though. You know, like, Beers of D2O.
Is there any chance that this occurs in nature in larger concentrations that might be usable by an organism anywhere?
So I actually just read about this, but apparently a lot of comets have the isotope ratio that is found here on Earth. So it's like basically 155 parts per million deuterium to hydrogen. And so like one of the theories is that the water source that we have from Earth is actually from comets, because in most of the universe it's at a different concentration. But it's not inconceivable to think that there are different concentrations around the planet. And I was actually talking to somebody today that it might be interesting to do like deep ice core samples and test the D to H concentration then, and you can probably see what the ratios were in different parts of the world at different times, you know, from back in the past millions of years, billions of years, last week.
Well, I was just imagining that, I don't know if you guys know the shadow biosphere stuff, the astrobiologists going on about it. The idea that there's this second form of life hiding somewhere on Earth that we haven't discovered yet. You know, perhaps using a different general organization, maybe not even DNA. But I was just imagining them all running around hopped up on D2O.
They'd probably like drink that stuff like cereal.
They're Gatorade. No one drinks cereal.
What do you mean? Well, yeah, nobody drinks cereal, but still.
You know, scientists are accused of having odd failings, but
the best deuterium cereal would be called D2Os.
part of a healthy breakfast. Nice. Wow.
Yeah.
Okay. Heidi wins for best bad joke.
I was going to say something, but I didn't want to hurt her feelings.
Oh no, that was the lamest joke ever. But we award points for that.
That's okay.
So we should. I should actually point out at one point for the people who haven't actually caught on to this yet. Anthony is actually our first physicist guest, and not necessarily a biologist.
I would say from your talk today, biophysicist.
Right.
Yeah.
Okay, biophysicist. All right, we're still straddling the borders.
I don't know if I'm allowed to say this now because I did just get awarded my Ph.D. Are they allowed to take that back? Because I haven't really thought about like real physics in like 3 years.
I don't think it's common to take it back, unless of course you copied all this from somewhere else.
It would be very easy for people to figure out if I copied any of it, since it's all public.
So let's actually talk about that. So you're a proponent of the open notebook idea. So what does that mean to you?
The simplest explanation I have is that I publish all of my results and methods in real time as I do the experiments. And so literally everything that you've seen on my notebook was published like seconds after I finished that part of the process. Or even like the data is like, I just made this graph, it goes on the notebook.
Did you?
How much has that feedback from that, influenced like the direction of your project?
There's been a couple times where I'll like get stuck on something and like all of my notebook entries go to Twitter automatically. So like sometimes I'll just literally ask a question as the title with no subject, and somebody will reply to me on Twitter within minutes, like giving me some new direction or something new to think about. And actually one of the scientists who comments frequently, his name is Bill Hooker, who's just been really interested in the stuff that I've been doing. He's been providing me like the most feedback. And actually, when I was talking about the tardigrade stuff earlier, one of like the world experts on tardigrades randomly found my notebook, and he just started like commenting on like extraction protocols and what to look for, what the eggs look like, what the tardigrades actually look like in the water, which I thought was really valuable feedback. I'm like, here's this random physicist who has no idea what he's doing, who just decided to go for a hike one day, and he is like trying to learn this on his own, and here's like a world expert like giving him really valuable feedback.
So tell us what your website is again?
Oh, it's research.iheartanthony.com.
Well, we all heart you, Anthony.
Thank you very much.
How did you come up with I Heart Anthony? Besides just like, are you just born in love with yourself?
Narcissism.iheartanthony.com.
No, originally it was because like I'm a graphic designer also, and so I had this I wanted to have a graphic design firm, and I was like trying to figure out a name, and everyone's like, well, you should name your graphic design company kind of after yourself, but I also wanted it to be distinguishable from everybody else. And so I was like, okay, I want to have really good design work, and I want people to love my design work. So I basically wanted something that, you know, you could say to somebody, I heart Anthony's design, or I love Anthony's design. And so that kind of became the idea. So I went to sleep one night, and that name popped into my head, and I was like, it's perfect. I'm going with it. And then I decided that the website itself should be like this multidisciplinary thing where I talk about design, photography, and then science, which is also the most important thing in my life. So I just added the research part to the front of it.
So I guess I'll go ahead and ask the obvious question. Like, as you're doing this, like, were you at any point afraid of being scooped, or that anyone would, you know, grab your partial results and take it a step further before you could, or anything like that?
Not really. I've always been kind of just like sharing with my information, so I just had that mentality going into it. And as far as like being scooped goes, because I don't have a lot of expertise in a lot of the fields that I was doing, to me it was like if somebody could take this to the next step faster than I could, it was better for science overall. But it turns out not many people have ever really thought about deuterium effects on just things in general, let alone life sciences. So it kind of was to my advantage that I could share this openly with everybody and kind of generate interest in it. And not many people turned out really cared at the beginning.
But kind of related to that, like, was your advisor aware that you were doing this, and how did he feel about it?
Yeah, he was actually, he was the one who initially introduced me to the idea of open science. He came from one of the national labs where it's not really encouraged to be outgoing with your research, and he also came from a lab at Cornell as a grad student, where he was basically like forced to lie to other people to prevent scooping. It was like this really stressful environment, like absolutely crazy. And like I would love for him to like publish the stories that he's told me about this stuff. And so he was like, you know, when I first met him, he was like, okay, we're gonna do this. Like, we're going to be good scientists, but we're also going to do this openly, and we're going to share our research. And I was actually like, that's not how science really is done. That's mind-boggling. So I was like on board from the start.
Yeah, you know, I've been moving towards the open notebook idea myself, and but I've noticed that there's a definite, like, there are some segments of the scientific community that are very much more skeptical of the concept, and I find it tends to correlate with how crowded an area is or how competitive it is. I mean, like, you know, say ecologists seem kind of okay with the idea, but there's a lot of room in ecology and, you know, behavioral ecology. Like, you can come up with new ideas easily, and you're not really locked in races with other labs very often. You know, but say molecular biologists who are all working on something in cancer, I don't see them doing it anytime soon because they're so competitive that, you know, like they're literally racing to be the first lab to publish X or Y or Z. And I don't know how that's going to roll out. I think it's a good idea, and I'm trying to transition to it myself. I'm bogged down a little by the fact that I have collaborators who aren't into the idea yet.
Yeah.
But moving forward, I'm going to transition to it.
So I actually have two things that deal with that. One is I've worked with collaborators who are actually completely anti-open, and they needed me to do certain things for them, and I was like, well, I'll help you, but as long as you let me be open about my part of the process. I won't share anything that has to do with your aspect of the collaboration, but you just have to let me like write about the protocols that I'm doing or the potential use for this without interfering with your research. And so they were pretty accepting of that. But the other thing is that I think that the culture is eventually going to change, in that, you know, once, like, Figshare is a big part of this because they're allowing scientists to cite data sets. And so, you know, if everybody has this open notebook, you can just, like, literally in your publication cite, you know, this method comes from this reference, or this data set that I used here to corroborate the results come from this reference over here. And so, like, as long as there's ways to cite people as opposed to just strictly from a publication point of view, I think there will be more incentive to share your data because you're getting the same credit for sharing bits of your information as opposed to, you know, sharing all of it in one final publication.
Do you think this is going to get wound up in changing publication models for like journal papers? You know, we're seeing a seismic shift in how things are published these days. Do you think this is going to go in hand with open science?
I think it is. They're already starting to kind of change that in the first place. I mean, open access is becoming a major deal. Like every single publisher has some kind of open access journal that scientists can pay into. There's new open access publications that make open access cheaper to attain. But then there's also like publication companies like PLOS that are kind of like always looking for new ways to allow scientists to be open and encourage the openness. And now they just partnered with Figshare as kind of this like allowing you to directly cite data sets, even though I guess you could anyways. And they also started working with Science Exchange, doing the reproducibility initiative to kind of like encourage reproducing results as a method for scientists to get publications.
It's cool that, I mean, now that we have all these tools where, well, you know, like we were restricted by the printing process to have science boiling down to what you could fit into those pages, right? And it's really, I'm really interested in seeing how people and publishers and how everyone's going to deal with, you know, science becoming more immediate and going beyond these things. So that's really cool.
I'm going to ask a slightly devil's advocate question, not because I think it's true, but because I think we have to have these conversations. And there are some people who think that, you know, opening up science in this way will in some way negatively impact it, say diluting it, or, you know, it'll be too hard to separate the signal from the noise. There'll be too many people claiming to do science, and, you know, it'll be too hard to find the real stuff. And I've actually had conversations like this with people, right, who feel this way, who think that openness in science is actually going to harm science. And so you've thought a lot about this, and what would you say to somebody like that?
I think there already is a lot of like signal to noise, and I don't even mean it like that. There's like literally thousands of publication platforms. There's like thousands of journals and thousands of articles. It's impossible for any one scientist to keep up with all of the literature. And typically we all ourselves look into like our competitors, our collaborators, and then like the outlying field from there. So like basically you pay attention to like your innermost circle, and you pay selective attention to the next circle, and you pay very little attention to the circles in other fields. And so I think that, you know, if like, let's say in this world where everybody has an open notebook, you're pretty much going to know who the community is. You know, it works on Facebook and it works on Twitter. You know the circles of people that you're trusting the information from, and I think it'll eventually kind of evolve toward that kind of a model, as opposed to just like surfing Google Scholar for, you know, the latest hit, or you surfing Nature in general just for the latest publication.
But I think those circles are peer-reviewed. You know, like maybe a couple years from now, you start to have people, I don't know, that have their own, that are like doing science in their garage and want to have their own open notebook thing. And I'm not criticizing those people, but I wonder if, you know, in 5 years, if you have like your site that's part of like whatever university, you know, like I have my own research page at UT Austin from my lab and whatever, and that's how people can put the confidence and trust in the sort of things that I'm doing. Because I know if someone who has like a garage lab, I'm not saying, you know, anything's bad with that, but if they're showing me their PCR results and their gel, I'm not sure I trust that the same way that I would someone from a lab. Sure,
that's like a valid fear. But like, there isn't a single person that I know who wouldn't scrutinize even a peer-reviewed publication. So in that sense, like, why not put that same level of scrutiny to a non-peer-reviewed, like, you know, anything in my notebook, for instance. And also, the opposite of that is that in my notebook, you know, I allow commenting. So anybody could post, you know, like, oh, you did this method wrong, or I don't think that this data set is reliable as you're saying it is. Like, there could be essentially open peer review in my own notebook. And so as a visitor to the site who might use the resources, that you could see that, oh, there's been this argument here, I trust this source, or a whole lot of people here have used this resource for beneficial purposes, so I trust this more.
And so kind of in that note of like trustworthiness of the data that you're putting online, like I could see putting everything online influencing how I do things in two ways, right? For one side, you might want to get, you know, immediate stuff out as fast as you can and make it available for people to discuss and give and weigh in. On the other hand, since you're exposing yourself since the beginning, you might end up being really careful about the quality of what you show, and like filtering it several times before putting it before showing it to people that will be judging it, right? So did you feel any of those two types of pressures when you were posting your stuff in, or you're just going as it went?
I actually do both, right? Like I'll publish something as soon as it happens, and if I'm not confident in the results, I always add like a little note, like this is what I did, and these are the results I got. I'm not really sure if I trust these results, so I'm going to do another trial tomorrow. But the other thing is that, you know, I make sure that when I'm doing the stuff in the lab that I do it to the best of my abilities, because I know I'm going to have to post about it later, and if I hide anything, I'm positive that it'll be discovered at some point in time. So if my methods aren't consistent with my results, then, you know, I don't want to be discovered as a fraud. That'll ruin my entire career. And so for me, it's just better to, you know, not only just be open, but also the openness pushes me to be a better scientist.
Yeah.
So you feel it adds a layer of accountability.
Yeah. Oh, it totally adds a layer of accountability.
You know, I was interested in that question that Heidi asked, actually, because it kind of signals that interface between the professional scientists and, you know, the amateur science community, which in biology is actually coming up hugely. I mean, it's always been there in, you know, in the higher organismal levels, and, you know, beetle collectors and, you know, amateur taxonomists and so on. But even at the molecular biology level, it's starting up with DIY bio and open PCR and, you know, all of these initiatives to allow people to generate data on their own. And I wonder if the open notebook concept is going to play a part in maybe there being a revival of that sort of amateur science, you know, in biology and elsewhere.
I think it would be pretty cool, honestly. Like, I don't have a problem with people in their homes doing experiments. I mean, if like a little kid is doing a project for his science fair project and he's publishing it openly, I think it would be really impactful for him in his youth, or her in her youth, to have a real scientist find this information and then like really comment on the stuff, or like, you know, give them feedback and improve their methods, and like work with scientists from around the planet one-on-one to develop stuff, especially for amateur astronomers. I mean, I don't know if you've heard of Galaxy Zoo, but they allow anybody with a computer to categorize galaxies. And so to have actual astronomers, professional astronomers, and amateur astronomers working together in the forums to classify stuff actually makes everybody overall more interested in science, which I think is the biggest aspect of all of this open science stuff anyways.
Well, that's a very positive note, and I think we can wrap it up there. It breaks my heart because I'd like to talk about this for hours with you, but we've reached the end of our time, and maybe we can have you back one time and talk about it some more.
That'd be awesome.
So if people want to know more about your stuff, they can find you on Twitter at the Science of Ant.
Yes.
And research.iheartanthony.com.
Mm-hmm.
And of course, if you want to find out about us, we're at breakingbio.com. And I want to thank Anthony for coming on today.
Thank you for having me.
Yeah, congratulations. Yay.
Yeah.
Yay.
I'm sure you drink one for each of us.
And all the audience people as well.
Yeah.
So,
All two of them. Yeah.
Yeah. So that'll be 4 in total. Good luck.
I'll have 2 for everybody then.
Excellent. And alcohol poisoning. So, but thanks for joining us, and we'll see you guys next time.
Thank you.
okay.
Congrats and
Bye.
thanks.
Thank you. Bye.